Chapter 4 is on tissues. There are 4 types of tissues, but in this chapter, we are going to concentrate on epithelial and connective tissues proper. Epithelial tissue covers and lines. It is avascular, meaning no blood supply. It is innervated, meaning there are nerves. And it's highly cellular, meaning many cells with little space between. Epithelial tissues are always resting upon a basement membrane with CTP on the other side. Epithelial tissues are said to be polarized. That means that different borders have different specializations. The free or apical border is on top, lateral on the side, basal on the bottom. Basal borders are always resting on a non-living structure known as the basement membrane. Some apical border specializations include the movable cilia, which are composed of microtubules, or microvilli. Microvilli do not have microtubules, they simply increase surface area. This is a diagrammatic of a cilium with its 9+2 arrangement of internal microtubules, and again, it works to move things along. Microvilli are simply extensions of the plasma membrane. They do not have microtubules, they simply increase surface area for absorption and secretion. Basal border features include a basement membrane, non-cellular, 2 parts to it. Basal lamina would be a product of the epithelial cells. On the other side would be a layer of reticular fibers. Those are products or synthesized by cells of the connective tissue proper. On the lateral borders, there are several specializations that we will see in greater detail in the next slide. From top to bottom, we have tight junctions where the proteins of adjacent membranes fuse. Tight junctions go around the entire perimeter of the cell and provide a permeability barrier. Desmosomes, tiny binding bodies, are like little buttons that strengthen cell-to-cell adhesion and binding. Gap junctions include connexons, which are essentially proteins that span from one cell to the other. These proteins, connexons, have internal channels that allow for cell-to-cell communication. This slide 10 shows all of the features that we have just discussed. Classification of epithelia would be twofold, either by according to the row of cells. One layer would be simple, more than one would be stratified. Shape of the cells would include flat, squamous, cube-shaped cuboidal, tall would be columnar. Goblet cells are single-celled exocrine glands that produce mucin. Mucin combines with water to produce mucus. Goblet cells are often sprinkled in between epithelial cells. Specific types of epithelia would start for our purposes, we'll start with a simple squamous epithelium, the thinnest epithelium, one row of flat cells. This is for diffusion and filtration. Location of simple squamous epithelium would include things like the air sacs in the lungs. Those are called alveoli. The lining of the heart, lining of blood and lymph vessels as the endothelium, and is a component of serous membranes as the mesothelium. Glomeruli of the kidneys are also simple squamous. Don't worry about the last one until we get to the kidney. Simple cuboidal epithelium is very common in kidney tubules, not glomeruli, kidney tubules. These cells are specialized for absorption and secretion. Simple columnar epithelium would be found in the digestive tract. In this case, there would be microvilli to facilitate absorption and secretion. Uterine tubes in the uterus, the simple columnar epithelium is ciliated to help move things along. Stratified squamous epithelium, many layers, top cells are flat. Two types of stratified squamous epithelium: keratinized with the waterproofing protein known as keratin, that would be in the epidermis. Non-keratinized without keratin would be lining of the mouth and lining of most of the esophagus. Transitional epithelium transitions in shape from thin to thicker depending on the volume of the structure that it's lining. And it would be found in places like the urinary bladder, the ureters or ureters, and the urethra. Again, transitional epithelium transitions in shape from thin to thick to accommodate for volume. Pseudostratified epithelium is actually one row of cells. However, some cells are short, some are tall. They may have goblet cells scattered in between. A very common location for pseudostratified epithelium would be in most of the respiratory tract where it would be ciliated. A little bit about glands, not too much. Two types of glands. Endocrine are ductless glands that secrete hormones. That's the only product, the only thing they secrete. And these hormones are secreted directly into the bloodstream. The example here would be the cells within the pancreas, the cells of the islets of Langerhans or pancreatic islets, which secrete various hormones, including insulin, into the bloodstream. Exocrine glands secrete through a system of ducts. Their products of secretion would include things such as digestive enzymes, sweat, sebum, which would be oil, and others. Last slide will show you different types of morphologies of multicellular exocrine glands. Concentrate on the morphology or structure of the secretory units depicted in a pinkish color here. The ones that look like a tube are called tubular. The ones that are rounded and look like grape clusters are called alveolar. Very often you will have a combination of both. In that case, it's referred to as compound tubuloalveolar.